Welcome, friends, to another exciting episode of AP Biology Unit 4. Today we're going to focus on the regulation of cell cycle. And we're also going to talk about what happens when a cell loses its ability to regulate its own division. Cell division out of control is cancer. And we all want to know more about cancer. It's something that strikes fear in all of us, I think. So this is a fairly interesting and somewhat complex topic. We're going to be discussing the information found on pages 192 to 197. So let's get started. The first thing you need to know is that cells vary a great deal as to how often they divide. How long it takes to do a complete cycle, G1, S, G2, mitosis, cytokinesis, how often and how fast that process takes place varies a great deal from one species to another, and within your own body it varies quite a bit from cell to cell. Skin cells, for example, must divide very rapidly and very frequently because you lose millions of skin cells every day. They die, you have to replace them. Red blood cells live up to 4 months. So the cell division process that takes place within your bone marrow to replace those lost red blood cells happens a little bit slower than it does, say, in the skin. Liver cells will divide rapidly for short periods of time if you are in need of repair or regenerating malfunctioning liver tissue. Those cells can divide rapidly, but they can also go long periods of time without dividing. And as we've previously discussed, once you reach adulthood, your brain and spinal cord cells, many of them at least, slip into kind of a permanent G0 where the cells continue to live and function, but they just no longer divide. They no longer participate in cell cycle. So how exactly all of that is regulated and how the cell controls how quickly and how often it does cell cycle is quite complicated, and I'd be lying if I said that scientists have all of this figured out. They don't. They're still working on learning exactly how this works, but there are some things that we do know and that I can share with you today. I can tell you that the stages of cell cycle, G1, S, G2, and so forth, are controlled by a very complex network of chemical signals. Some of these signals come from within the cell itself, and we're going to talk about a couple of those signals today. Sometimes there are signals outside of the cell, signals that may be coming from other nearby cells that will stimulate a cell to begin dividing more rapidly or more slowly. You should also know that there are several, at least 3 that we know of, checkpoints, sort of safety roadblocks that are built into the cell cycle so that typically a cell is only permitted to complete the cell cycle process if it is a healthy cell with non-mutated DNA. We'll talk about that in just a moment. Speaking of how fast cells divide, I can tell you this, that there's probably never a point in your life where your cells are dividing any faster than they did in the first few hours of your life. What you're looking at in this time-lapse illustration is the very beginning of your life. We all started out as a single-celled zygote. That's the cell that was formed when the sperm and the egg fused together. And that cell, that single cell, began doing cell cycle. G1, S, G2, mitosis, cytokinesis. And it cleaved or divided into 2 cells. which quickly divided into 4 cells. And then here's what it looked like when you reached the 8-cell stage. At this point in your life, every one of these cells were identical. They had not specialized yet to become skin cells or muscle cells or kidney cells or whatnot. They were just cells, genetically identical cells produced by mitosis. And of course, those 8 cells divided to make 16, which divided to make 32 and 64 and 128. And at some point, you sort of developed into a hollow ball of cells. You see it was kind of hollow in the middle. And then some of these cells on the outside started to fold inward to form what would eventually become your digestive system. You see this little pouch right here? That's what became your stomach and your intestines. And wow, such a cool process, but it really started out with just rapid mitosis. In fact, in these early days of your life as a developing embryo, the mitosis is happening so fast that there's no time for G1. Notice the ball of 8 cells is no bigger than the original 1 cell. And even though this is obviously a magnified view, these pictures are drawn to scale. The 128 cell stage, this ball of cells that's made up of 128 cells, was no larger than the original cell. And that's because these cells were dividing so fast that they didn't really have time for a G1. There was no growing taking place in between cell divisions. Now, obviously, at some point that started to change, and the cell cycle process did slow down a bit, especially as your cells began to specialize to form your various tissues and organs as you developed from a ball of cells into a fetus and eventually something that we would recognize as a baby human. So this was the point in your life where cell division was happening very rapidly indeed. I mentioned a few moments ago the existence of several checkpoints that we know about within the human, well, not just human, but within cell cycle, the same cell cycle that all animals go through, and indeed plants as well. And the most important of those checkpoints is called the G1 checkpoint. It's also sometimes called the restriction point. And if you think about it, if you're going to stop cell cycle and find mistakes in the DNA and fix those mistakes, make corrections to those mutated genes, the best time to do that is before S. Because what happens in the cell during S? Do you remember? That's right. The DNA is copied. Every strand of DNA is copied during S. Well, we don't want to copy the DNA if it has a mistake in it. So the G1 checkpoint is where the cell has just about finished growing, and it's about to begin replicating the DNA, and it just kind of pauses. Various enzymes will check for DNA damage to see if any mutations have occurred. And if there are some mistakes in the DNA, very often those can be corrected. You actually have an editing, proofreading mechanism that operates in the nucleus that can fix most mistakes. If a mistake exists, but it is not detected, then the cell will proceed past the G1 checkpoint. The DNA will be copied, including the mistake or the faulty DNA. That will be copied as well. So now we have 2 copies of that mistake. And we don't want both of those copies of the faulty DNA, the mutation, to be passed down to the 2 daughter cells. So there's another opportunity to stop and detect and fix those mistakes before mitosis. This is called the G2 checkpoint. It's right before mitosis begins. So that would be another time that it would be really important to try to catch any mistakes and fix them. And then about halfway through mitosis, maybe somewhere around metaphase, there's another checkpoint where one last time perhaps we can catch the mistakes that we've missed at the two previous checkpoints. And before that cell splits into two cells, hopefully we can fix those mistakes. Again, the most important checkpoint is the G1 checkpoint because that's the chance to fix the mistake before it's copied. At the top of this lesson, I said that one of the things we will discuss today are the chemical signals that help us regulate our cell cycle and our cell division. And this particular part of cell division is pretty complex, and this is the part that scientists have not quite figured out how all of it works, but there are some things that we do know. For example, we know that there is a protein that is always present within our cells, and it's called MPF, which has about 3 different names. It really depends on what textbook you look at. Sometimes it's called M phase promoting factor. Sometimes it's simply referred to as mitosis promoting factor. Basically, this is the protein that is sort of the go signal that is going to actually trigger mitosis to start. Notice MPF is always present within the cell. It is a protein, it's an enzyme, and it's always present in the cell. It is not always in its active form. So MPF kind of has an on switch and an off switch. And when the off switch has been flipped, it's not able to do its job. It's not able to signal the beginning of mitosis. MPF is part of a group of enzymes that are called Cyclin-dependent Kinases. Whoa, what does that mean? Well, the first part means that MPF is only active, it only switches on when another chemical is bound to it. And that other chemical is called cyclin. Okay, so let me bring this up. Cyclin is a separate protein that is produced in the cell, and cyclin is not always present within the cell. MPF is always present, cyclin is not. The concentration of cyclin fluctuates. It goes up and it goes down. So there's going to be times where the cell produces lots of cyclin. There's going to be other times when the cell does not produce cyclin. Now, cyclin's job is to bind to MPF because MPF is cyclin dependent. It can only do its job when cyclin is bound to it. And MPF is also considered to be a kinase. What does that mean? A kinase is any protein or enzyme that kickstarts a chemical reaction or a process by providing energy. And typically the way that it does that is by taking an ATP molecule and breaking off a phosphate. Remember, that releases energy when you break off the third phosphate from ATP. And the kinase will stick that phosphate group onto some other molecule to give that molecule an energy boost and kickstart a chemical reaction. Okay, so let me back up. What are we saying here? We're saying that when your cell produces cyclin, cyclin has the ability to physically bind to MPF. So this chemical binds to this chemical. What does that do? It triggers MPF to change into its active form, and when MPF becomes active, it acts as a kinase. It adds a phosphate group energy to another molecule to kickstart mitosis. That's why it's called mitosis promoting factor, because it kickstarts mitosis. So during interphase, when the cell is not dividing, and it's not supposed to be dividing, interphase is when mitosis is not happening, the cell simply shuts down the production of cyclin. If the cell does not produce cyclin, then cyclin cannot bind to MPF, and MPF will be in its inactive form, so mitosis does not occur. But after G1 and S and G2, all of a sudden it's time for mitosis to begin. How does the cell trigger mitosis? By producing cyclin, lots and lots of cyclin. The cyclin will bind to the MPF molecules. They will switch to their active form. Mitosis begins. So here's a graph that kind of illustrates this. I've always thought this was kind of a confusing graph, but notice the red line shows the rising and falling of how much cyclin is in the cell. So when there's very, very little cyclin in a cell, the cell is in interphase, G1, S, G2. But towards the end of interphase, as we move into G2, you notice what's happening? The level of cyclin is rising, 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 rising. And when there's a whole bunch of cyclin, what are those molecules going to do? They're going to start binding to MPF. So the black line shows how active MPF is. When MPF becomes fully active, what happens? Mitosis begins. And when it's time for mitosis to finish and telophase is over, and now the cell is doing cytokinesis and it's splitting into two cells, all of a sudden the level of cyclin crashes almost to zero. MPF switches back to its inactive form. That's the falling of the black graph here. The black line goes down. MPF is inactive, and we go back to interphase. All right. So this is all a little confusing. You might want to back it up at this point and watch the last 5 minutes of the video a second time, because the interaction between cyclin and MPF is a complicated interaction. So normally, the process of cell cycle is heavily regulated so that cells have plenty of time to grow, copy all of the DNA accurately, and then carefully, through the process of mitosis, separate the copies of DNA and then divide the cell. And you learned about that in the previous lesson. But what about when the cell starts dividing out of control? What if there are a series of mutations within the cell that cause it to not be able to regulate its own division? That is cancer. So as I said, cancer cells are cells that are dividing out of control. And scientists have spent a lot of time studying the characteristics of cancer cells that are growing in vitro. In other words, if you do a biopsy and you collect a small sample of cells from the body, maybe from a tumor, maybe from an organ, and you grow those cells in the laboratory in a vial or on a petri dish, and you give the cells nutrients to keep them alive, and you study how they divide and how they behave, scientists have noticed that cancer cells grow in a very different way than healthy, non-cancerous cells. So we're going to talk about 3 characteristics that separate how cancer cells behave compared to normal healthy cells. Cancer cells have lost three characteristics that normal healthy cells have. And the names of these are a little confusing, but I'm going to give you some examples to explain. So cancer cells have lost density-dependent inhibition, anchorage dependence, and mortality. Now that doesn't really mean that if you have cancer you're immortal. It doesn't mean that at all. But that's just a term that's used by scientists when they're studying cancer cells. So again, these are three characteristics that normal healthy cells have that cancer cells have lost. So the first one was density-dependent inhibition. If you take normal healthy human cells and you remove them from the body and you grow them on a petri dish, a little glass tray with nutrients, they will divide and divide and divide until they cover the bottom of that petri dish. And when the whole bottom surface of that petri dish is full of cells and every single cell is touching the side of the dish or another cell, they stop. When there's one single layer of cells that covers that petri dish, they stop dividing. When they reach a certain density, they stop dividing. So that's called density-dependent inhibition. Cancer cells don't do that. Cancer cells will keep dividing as long as you provide them with nutrients. They will keep dividing and dividing. They'll pile up on top of each other. They will not stop dividing with one single layer. They'll keep dividing, forming irregular clumps and multiple layers of cells. The second characteristic that normal healthy cells have is called anchorage dependence. A normal healthy cell, when you grow it on a petri dish, it will only divide if at least one of its surfaces, one part of this plasma membrane, is anchored or touching a solid surface. So they like to be sort of growing on a surface. And once they reach a point where the cells have formed a single layer on the bottom of the petri dish and they have to start piling up on top of each other, they don't like that. And they don't divide anymore. So normal healthy cells do not pile up and form multiple layers. This kind of goes hand in hand with what we were just talking about, density-dependent inhibition. Cancer cells, again, have lost this characteristic. Cancer cells will continue to divide whether or not they can touch the bottom of the petri dish. As long as you give them nutrients, they will continue to divide and divide and divide, piling up on top of one another, forming irregular clumps. They're very different in their behavior compared to healthy cells. And finally, cancer cells have lost mortality. Now this is in quotes. We're not really talking about, we're not really talking about living forever here. But if you take normal healthy cells from the human body and you grow them in the laboratory and you let them, grow until they fill up the petri dish, and then you transfer them to a new petri dish. So now they've got plenty of room. They'll keep dividing again and again and again. And if they fill up that petri dish, you can transfer them to a third one. But then after 20 to 50 generations, they just stop. In other words, they'll divide. That's one generation. They'll divide again. That's two generations, again and again and again. After 20 times, or maybe 30 or 40, but no more than 50, they'll stop. And it's very difficult to keep healthy cell lines growing in a laboratory because they don't like to divide. Once they get outside of the body and they're separated from all of the signal molecules that are produced by the body, they really don't like to divide very much. Cancer cells have no limit. They will divide and divide and divide as long as you keep providing them with nutrients on a petri dish. They will divide for weeks, months, years. The most famous example of cells that have lost their mortality are called HeLa cells. HeLa cells originated in a cancerous tumor within the body of a young woman named Henrietta Lacks, who I believe lived, in Maryland in the 1950s. Miss Lacks had cervical cancer, terminal cancer. They were not able to offer her any medical treatments at this time. So she got a very grim prognosis from her doctor. But at some point at Johns Hopkins Medical Research Hospital in 1951, a doctor collected a biopsy from the tumor, the cervical tumor in Henrietta Lacks's body, and he began to study those cells under a microscope. And he discovered that even as cancer cells go, these cells were remarkable in how aggressively and how rapidly they were dividing. And they seemed to divide and divide with no end. As long as he transferred cells from Henrietta's tumor to a fresh petri dish with fresh nutrients, they would continue dividing and dividing. Pretty soon, at Johns Hopkins Medical Research Hospital, they had vials filled with Henrietta's cells, or at least, clones of her cells that had been formed by the cell division of her cells. And pretty soon, Johns Hopkins began to share some of these HeLa cells. Again, they came from Henrietta Lacks. That's why they're called HeLa cells. Other research institutes began to use HeLa cells in their research. At some point, Johns Hopkins began charging for sharing these HeLa cells and shipping them all over the world to research institutes. And almost every major advance that's been made since the 1950s, so we're talking 70 years, every major advance, every major discovery that's been made in cytology and certainly in cancer research has been done with HeLa cells. They are everywhere. They are the most studied cancer cells in history, and every major research lab in the world today uses HeLa cells. So even though Henrietta Lacks has been dead for almost 70 years, and she died fairly rapidly after her cancer diagnosis, her cells, or at least the descendants of her cells with her DNA, are still dividing, and they're still living. So they have certainly lost this mortality limit. Remember, a normal healthy cell will only divide for about 20 to 50 cell generations, a few days at most, and then they stop dividing. This is such a fascinating story. One of the sad components to this story, besides the fact this young poor woman died from this horrible cancer, is that her children and her husband, and her husband was a crud of a human being, it turns out. He was very abusive and cheated on her and was an awful man, but neither he nor his children with Henrietta nor any of their descendants ever knew, or for a long, long time at least, that Henrietta's cells were being used for research. Nobody ever asked the Lacks family, hey, can we use Henrietta's cells? Can we start selling Henrietta's cells? Can we make millions and millions of dollars developing cancer treatments using Henrietta's cells? And so one of the most interesting nonfiction books I've ever read came out probably about 10 years ago, written by Rebecca Skloot, who is a science writer. And the book is called The Immortal Life of Henrietta Lacks. It tells the story of her life and how her cells became famous. But it also tells the story of how this poor, uneducated family, many of them are still poor and many of them are uneducated, how they have slowly come to realize the importance of their ancestors' cells, and how still to this day the family is not receiving any compensation. They're not being paid for this tremendous contribution that was made to medical science without the knowledge of this poor woman. I would encourage you to read this book. I guarantee you'll find it interesting and somewhat heartbreaking. And that is your extra credit phrase that pays: The Immortal Life of Henrietta Lacks. The Immortal Life of Henrietta Lacks. Okay, so some terminology that's often used when we discuss cancer. Here we're using breast cancer as an example, but all cancers kind of use the same terminology. A tumor is a mass of rapidly dividing cells that are irregular in some way. There's a series of mutations that has caused the cells to divide in an out-of-control way. Now, if that tumor is not invading surrounding tissue, if it's confined to a very localized area, we would refer to that as a benign tumor. And even though the cells are technically irregular and they are dividing out of control, if you are diagnosed with a benign tumor, you probably would not be considered to have quote unquote cancer. But when we go to the next image, you notice it's quite different. So this is a tumor, and just like the benign tumor, this started out as a single cancer cell that divided quickly to make two cancer cells, which divided to make 4, 8, 16. But now this tumor has begun to spread cells outward from the tumor itself, and the cancerous cells are dividing so rapidly that they are invading the surrounding tissue. This is going to be much more difficult to remove surgically. This is called a malignancy or a malignant tumor. And so that's, of course, a worse diagnosis and more difficult to treat. The worst case scenario when it comes to cancer is when a tumor develops and grows to the extent that some of the cancer cells, in this case from a breast cancer tumor, begin to break off of the tumor and spread into lymph nodes and lymph vessels, and sometimes even into blood vessels. And once those cancer cells get into the lymph and the bloodstream, they can travel to other parts of the body. So this is how breast cancer becomes bone cancer or lung cancer. You know, when you talk about cancer spreading, the medical term for that is metastasis. Cancer cells separate from the tumor and move to other parts of the body via blood and lymph vessels. Again, the term for that is metastasis. So how does this all begin? Well, cancer cells start out as normal, healthy cells, but as cells divide, you know, the cells throughout our body are constantly dividing, some more rapidly than others. But during the cell division process, and especially during S, when the DNA is being copied, sometimes mistakes happen. We call these mistakes mutations. Not all mutations lead to cancer. I don't want you to get that idea in your head. Not all mutations lead to cancer. But if a cell has a specific series of mutations that affect the genes that are in charge of regulating cell cycle, so it's usually multiple mutations, not typically just one, but if there's multiple mutations affecting the genes that regulate cell cycle, you now have a cell that is no longer a normal cell. It has transformed into a cancer cell. Now that's still just a single cell. Somewhere in your body, you have 30 trillion cells. So one cancer cell is not all that alarming. And this is not pleasant to think about, but you probably do have some isolated cancer cells in your body. We probably all do. And the older you are, like me, the more likely you are to have these cancer cells. But they don't usually live very long because very often the immune system will detect that there's something unusual about this cell. It's not a normal cell. Something's wrong with this cell, and the immune system will kill it. And that's wonderful, right? You want the immune system. That your immune system produces a type of cell called a cytotoxic T cell, and that's one of its main jobs, is to attack cancer cells and kill them before they have a chance to divide and produce a tumor. Now, if that doesn't work, another possibility is you have a series of genes that you inherited from your parents. They're called tumor suppressor genes, and one of the most famous ones is called p53. A little bit more about that in a moment. And p53, this tumor suppressor gene, can kickstart two separate mechanisms which can block a cancer cell from becoming a cancer tumor. Now, if both of these fail, if the immune system fails to destroy the cancer cell, and if your p53 activity doesn't kick in properly, then you're in trouble. Because then that one cancer cell is going to divide by mitosis to make two cancer cells, and they'll divide to make 4, and they'll divide to make 8. Now it's out of control, 16, 32, 64. Pretty soon you've got a tumor that has formed by rapid out-of-control mitosis. Hopefully it's a benign tumor, but if it continues unchecked and untreated, it's going to become a malignant tumor and, of course, eventually metastasis, and that's not the pathway we want to go down. So let's talk about how p53 can help protect you from a cancer cell becoming a cancer tumor. p53 is the name of a gene. It's found in all vertebrate genomes, so this is not just a human gene. This was passed down to us from our reptile ancestors, and it was passed down to them from their amphibian ancestors, and it was passed down to them from their fish ancestors. p53 is a gene. It codes for a protein called p53 protein. No surprise there. What does the protein do? p53 protein controls the expression of many other genes. So wow, this is kind of mind-boggling. So p53 is a gene that causes your cells to build a protein, and then that protein, that p53 protein, acts like a switch, and it can flip on the switches for other genes, and those other genes can kickstart regulatory mechanisms to control cell cycle, stop cell division temporarily, and even repair the mistakes that are in the DNA. Sometimes it's possible to fix a cancer cell, repair the DNA damage, and it's no longer a cancer cell. Isn't that wonderful? So p53 is considered to be a tumor suppressor gene. Let's see how it works. So here's a cancer cell that has sustained DNA damage. It has probably multiple mutations. And so this cell now has the potential to divide out of control to form a tumor. How can p53 save us from that? If the cell has relatively low levels of damage to the DNA, in other words, if this cell is fixable, p53 protein will kickstart a series of chemical reactions that will cause the cell to temporarily arrest its cell cycle. In other words, it kind of goes on pause. This cell will temporarily stop dividing until other enzymes come in and repair the DNA damage. In other words, It's not a cancer cell anymore because we have fixed the mistakes in the DNA. And once those mistakes have been fixed, the cell is allowed to re-enter the cell cycle and continue happily dividing, making normal, healthy cells. And that's a wonderful thing, that you're literally taking this cancer cell, fixing the mistakes. Once it is no longer a cancer cell, no longer a risk, you allow it to divide. Now, the other possibility, if we have a cancer cell with high levels of DNA damage, the p53 protein will kickstart a series of events that results in apoptosis. That's literally cell suicide. And this may involve lysosomes within the cell rupturing, spilling out their hydrolytic enzymes. It's maybe more complicated than that. We don't know all of the details yet, but somehow p53 protein triggers this cell to kill itself. And hey, that's a good thing. If it's a cancer cell and it's not fixable, we want it to kill itself. And that's what apoptosis is. Unfortunately, some of us, some humans, some animals, have inherited from their parents a mutated version, and sometimes two mutated copies of the p53 gene. If you have a mutated p53 gene, it's not going to function properly. It's not going to code for the p53 protein, which means neither of these two things are going to happen. So with a mutated p53 gene, this cancer cell has nothing to stop it from dividing and forming a tumor. I say it has nothing to stop it. That's not really true, because remember, the immune system, we talked about that earlier. Let me go back. The immune system could still detect that something's wrong with that cancer cell and kill it. But whereas a normal person, most of us have both of these protective mechanisms, a person who inherited two mutated copies of p53, they don't have this as an option. So the only thing that can protect you from cancer in that case would be your immune system. So let me go back. Notice over here it says wild type p53. What the heck is that? Wild type is a genetics term. It just means normal p53. That's what you want. That's what you hope you inherited from both your mom and your dad. Normal wild type p53. And if you did, whenever a cell becomes a cancer cell, it will either, the p53 protein will either fix the damage and allow the healthy cell to divide, or p53 protein will trigger apoptosis and the cancer cell will kill itself. If either of those two fails, or if both of these fail, that's probably because you inherited mutated p53, and so it's going to be easier now for that cancer cell to divide and form a tumor. So one last point about this. If you did inherit two mutated copies of p53, one from your dad, one from your mom, does that automatically mean you're going to have cancer someday? No. It means you're more likely to develop cancer, but it doesn't mean you will have cancer, because there's no guarantee that any of your cells are going to become cancer cells. And even if they do, there's a pretty good chance that your immune system can still take out and destroy that cancer cell. Okay, before I bring to a close this already very long lesson, let me shift gears in a major way, and let's talk about something that has absolutely nothing to do with cancer and has nothing to do with mitosis, but this is a type of cell division. I want to very briefly talk about binary fission. Binary fission is a simple version of cell division that takes place in bacteria. prokaryotes. Bacteria do not do mitosis like our cells do and like plant cells do, for the simple fact that a bacterial cell does not have a nucleus. Remember, there's no nuclear envelope. It does not have multiple chromosomes that have to be copied and separated. It only has just one chromosome or one piece of DNA. So this is a much simpler cell division process. It does not involve spindle fibers. It does not involve kinetochores and spindle fibers attaching to chromosomes and pulling them apart. None of that happens. But it does have some things in common with mitosis. So the first thing that happens is the DNA, the single chromosome inside the bacteria cell, has to be replicated. It has to be duplicated. And so what's happening between this picture and all the way down to here is that the amount of DNA just doubled. It's a little hard to tell from the picture, but the amount of DNA has now doubled. This cell had only one copy of its DNA. This cell has two copies of the DNA. And then what happens is the bacterial cell begins to elongate. You see how it looks much longer here? And as it elongates, the two copies of the DNA separate. They're not pulled apart by spindle fibers, but they do separate in a mechanism that's maybe not fully understood, but somehow they separate. And at some point, the cell cleaves into and splits to form two separate genetically identical bacteria. So it is like mitosis in that the DNA is copied and we have two exact duplicate copies of the DNA, and then when the cell splits, you end up with two genetically identical cells, just like mitosis. It's just a lot simpler than mitosis, and this process, again, in bacteria is called binary fission. Guys, that's the end of my lesson. I hope you enjoyed it. Don't forget to email me with the extra credit phrase that pays. The first 10 to do so get some extra credit. I hope you have a great day.